does this happen? How does one cell produce a second cell with the same exact DNA sequence? In other words, how does DNA replicate? This is a fundamental question that is the basis of life. We all come from a single cell that will produce an entire organism with trillions of cells that all have the exact same DNA sequence. And we are able to have children that share our DNA because our eggs and sperm carry our DNA sequence. Understanding how DNA replicates and how all of our cells can carry the same DNA sequence is the basis for understanding development, inheritance, heritability, and life. In 1953, the question lingered, how does DNA replicate? There were a few theories, 3 actually. One of them was conservative replication, where a DNA molecule would get copied and make a second new red DNA molecule. The second was dispersive, where the DNA molecule would be cut at various parts, each of which would get copied here in red and reattached to produce 2 DNA molecules. And the third was semi-conservative, where 2 DNA strands would separate, and each one would serve as a template to copy a second red strand, thus producing 2 DNA molecules. But which one was it? Many scientists did not believe that 2 DNA strands could separate because of how strong a DNA molecule is, so they were skeptical of the semi-conservative model. The only way to solve this was to create an experiment, to tell the new DNA strand from the old one. So 2 scientists, Matthew Meselson and Franklin Stahl, had an idea to design an experiment that would be known as the most beautiful experiment in biology. Their findings are published in their article titled The Replication of DNA in E. coli, published in May 1958 in the Proceedings of National Academy of Sciences. These scientists used bacteria to answer the question of DNA replication, since it could be easily grown in the laboratory. They grew the cells in the presence of a specific type of nitrogen, which is found in DNA that would make all of the DNA very heavy. They used centrifugation, which can separate things according to their weight. Initially, all of the DNA in the cell was heavy and was at the bottom of the tube, since it was grown in heavy nitrogen. Then they started growing these cells in the presence of light nitrogen, so all of the DNA made in subsequent cell divisions would be lighter. After one cell division, the DNA was half as heavy, so half of the DNA molecule contained heavy nitrogen and the other half didn't. This is not in line with the conservative DNA replication model, which would predict that one molecule would be all light and the other all heavy. However, this is in line with the semi-conservative or the dispersive model. After 2 cell divisions, the DNA molecule was now either half heavy and half light, or entirely light. So this is now not in line with the dispersive model, which would predict that the DNA after 2 cell divisions would contain a mixture of heavy and light DNA. Instead, this experiment agrees with the semi-conservative DNA replication model. Every cell gets one old DNA strand and one new one.